I'd now like to bring up to the podium Stacy Cohen, who's from Children's Hospital of Philadelphia, known colloquially as CHOP, and she's going to talk about genetic counseling and the like. Come along, Stacy. Thank you. Oh, I'm going to grab my phone.
Thank you for the introduction. Good morning, everyone. My name is Stacy Cohen. I'm a genetic counselor at Children's Hospital of Philadelphia within our epilepsy neurogenetics program, and today I'll be presenting Deciphering the Genetics in KCNQ2. So first, I'll just start with a brief introduction to general genetics concepts. We'll talk about different types of genetic testing that may have been ordered for your child, how they're different and what they're looking for, and then we'll talk about different types of genetic variation in the KCNQ2 gene. So starting at the very beginning, the building blocks of our bodies, called cells, use our genetic material in order to grow and develop over time. And this genetic material, called DNA, is packaged into chromosomes. And each of these chromosomes contains many genes. When you unravel them and look at the code, there are different segments that we call genes that each encode proteins that our body uses. And you can also see on the screen here, this is what the chromosomes actually look like if you were to look at them under the microscope. The chromosomes do come in pairs. There are 23 pairs, so 46 in total. One from each pair comes from each parent. And chromosomes 1 through 22 are the same in everyone, and biological females have 2 X's, and biological males have 1 X and 1 Y. The KCNQ2 gene is found on chromosome 20, and because we have 2 copies of this chromosome, we also have 2 copies of the KCNQ2 gene. So thinking a little bit more about how these chromosomes work. So the chromosomes are the packages of our genes, so when we unravel them, we're able to look at the actual genetic code called DNA, and this is a 4-letter code that our body reads in order to make proteins like KCNQ2. So there is an intermediate step in which the body reads the DNA and generates mRNA, which is the code that is used to actually generate the proteins in our bodies. So the body reads through the mRNA code in order to determine the building blocks of the proteins which should be inserted and in what order, and these building blocks are called amino acids. So once the body has put all of these amino acids together in the correct sequence, this is then used to build the final structure of the protein that our body is using. So KCNQ2, as we've been talking about throughout the morning, is a potassium channel. There are many, many different potassium channels in our bodies, and we've talked kind of at length before about the structure of the KCNQ2 protein, but here's another cartoon that I'm sure you'll see again too, that just shows the structure of the KCNQ2 protein and the different segments. So we've talked about how the genetics work in our body. The DNA code is used to generate proteins. But when you actually go in to meet with a geneticist or a genetic counselor or your neurologist to talk about genetic testing, there's a lot of different things or tests that may be ordered in order to better understand your child's genetics. And they're each looking at the genetics in different ways. So the most kind of zoomed out approach to looking at genetics is a test called a karyotype. And what this is doing is looking at the overall structure of the chromosomes and the number of chromosomes that are present. So this is a test that would tell us if there were large missing or extra pieces of a chromosome, or if there were a rearrangement of the chromosomes where pieces of the chromosomes are where they should not be. Another test that's commonly ordered when trying to understand the cause of a child's developmental differences is a test that's called a chromosomal microarray. This is a more zoomed in version of the karyotype test, which is able to better detect smaller pieces of genetic material that may be extra or missing. And then other types of tests that are commonly ordered are called gene panels and exome sequencing. And these tests are similar in that what they're looking for are differences in the actual code of the genetics. So they're looking for small typos or misspellings in the genes rather than looking at the amount of genetic material, which is what the previous tests were looking at. So we have over 20,000 different genes in our body, each serving a different purpose. And a gene panel is selecting a group of those genes and that are associated with a given phenotype, like epilepsy or autism, and it's reading through those genes specifically and looking for small typos or misspellings in just those genes. Whereas exome sequencing is not just limited to a particular group of genes. We're looking at most or all of the genes to try and assess if this is, to try and assess if there are misspellings in the genes. And this is often a test that is ordered when there are many different genes that can cause a given symptom in a child, and we're trying to kind of cast a wide net. Other types of genetic testing that you may have talked about with your care teams are parental testing. So when we think about the inheritance of KCNQ2 DEE, most of these we expect to be what we call de novo, meaning parents undergo genetic testing to see if they carry the same genetic change as their child, and we expect that only the child carries that particular genetic change. And if this is the case, then we would not expect a family to have multiple children with that same genetic change in KCNQ2. However, there are exceptions to this due to a phenomenon called parental mosaicism, which we'll talk about a little bit more on the next slide, where parents may have a small portion of their body that may carry this genetic change that wouldn't necessarily pop up when we're doing initial screenings. And this can be kind of difficult to discern clinically because often parents who are mosaic may be unaffected, or they may have mild symptoms that wouldn't have otherwise flagged that we should be concerned that they have this. They may have mild epilepsy or learning differences, and knowing about this may change recurrence risk counseling. So this is a diagram to explain the concept of parental mosaicism. The idea is that in a developing embryo, a genetic change occurs in one of those early cells, and then as that cell grows and divides, all of the daughter cells from that original cell with the variant also contain the variant. And this may lead to that variant being present in a smaller segment of the body. So for example, if a parent has multiple egg cells or multiple sperm cells that carry a genetic change in KCNQ2, we wouldn't necessarily expect them to have symptoms as a result because that change isn't present in the brain. But then when that individual goes on to have children, they can pass on that genetic change to their child or multiple children. So conclusions in thinking about these general genetics concepts. There are several genetic tests that may be considered and you may have talked about with your physicians to reach this diagnosis of KCNQ2. And it's important to discuss parental testing options with your care team in thinking about family planning. So moving on to the next segment, which is how do these different genetic variants cause disease? Which is a very big question, but we're gonna attempt a simple answer. So we have our genetic code that our body is using in order to generate the string of amino acids that correspond with the KCNQ2 protein. And so we've identified that there's a change in the code and correspondingly change in the protein, but what is this actually doing? So when we think about genetic variants in general, again, when I say variant, and some people may use the word mutation, but we're all talking about the same thing. This is what we say is a change in a gene, like a typo or a spelling change in a sentence. And in general, we know that most genetic changes are not harmful. We all have genetic changes in our bodies that make us different from one another, but don't necessarily have direct implications on our health. And so, of course, we know that there are harmful genetic variants, though. And these are variants where we expect them to disrupt or change the way that the protein product is made. So missing or abnormal KCNQ2 protein as a result of these KCNQ2 variants changes brain functioning. So it's important when we talk about these different types of changes that we're very specific in the way that we name them, especially in the era of precision medicine. All of these different variants may have different implications in the way that we think about your child's care. So there is a standardized nomenclature that we use in order to name these genetic changes. And there's really 3 different ways that we name these changes. We have what's called the G dot, the C dot, and the P dot, and these are all different ways of naming the same change. So the G dot refers to the genomic position of a given variant, and the number refers to where that variant is occurring. And then there is what's called a reference nucleotide, which is the letter that is typically in that place. And then the greater than symbol means is changed to, and then what we call the variant nucleotide. So we would read this as saying there's supposed to be a G there, this was substituted for an A. The C dot is actually referring to the cDNA, so you can kind of think of C dot as referring to the code of the DNA. And again, this is saying where in the code is this genetic change occurring? It's occurring at the 881st nucleotide position. There's typically a C, there's a T substituted. And then lastly, the P dot is referring to the effect of this change in the genetic code on the protein itself. So we would read this as saying there's typically an alanine in a particular position, and this is substituted for a valine as a result of the change in the genetic code. And these particular amino acid positions correspond with different places in the protein, and knowing that place in the protein is important in how we think about potential functional impacts. So missense variants are the most common type of genetic change that we see with KCNQ2 DEE. And this is the type of genetic change where we have a substitution of a letter in the genetic code, which leads to a substitution within the protein itself. However, there are other types of genetic variation that we can see in KCNQ2. And these different types of variants are notable by when you look at the P dot. This is telling you what's happening on the protein level. So what we call a nonsense variant means that the protein is stopping too early. And that's often denoted by an asterisk. There's what's called a splice site variant, which you would be able to tell if there's a +1 or a -1 on the genetic report. And there's also frame shift variants, which are denoted in the protein with this FS star and then a number. All of these variants are typically associated with what we call protein truncation, where we expect the protein to be cut off too early. And this can happen with KCNQ2 DE variants, but typically that we see missense variants in this particular phenotypic group. So when we think about the effect of these different types of variants on the KCNQ2 protein, what I have here is showing those subunits where we have 2 units of KCNQ2 denoted in green and 2 units of KCNQ3 denoted in blue. And I'm showing 2 subunits here to denote the fact that we have 2 copies of each of these genes. So in the wild type state where there's no genetic variants in either of these genes, we see that both of these channels are able to come together and function properly. And then when we think about so-called loss of function, there are different levels of this. So when we discuss those truncating variants on the last slide, they typically lead to a lack of ability to form the subunit. So we have 50% that is functional. And then we have dominant negative, which we can think of as a more extreme version of loss of function. And what this means is there's a change in the KCNQ2 protein, But this change is not severe enough that it stops that protein from binding with KCNQ2, which is what it's supposed to do. So when this is incorporated into the overall subunit, when it forms with KCNQ2, it's still not able to work properly. So instead of 100% or 50% being functional, there's only 25% functional. And so this is why a general sense of why we think that there's these types of variants can lead to more severe symptoms. And of course, this is a generalization, but this is the way that we can think about this. And then there are also gain-of-function variants, as has been mentioned previously. But there are lots of different ways that this can happen that cannot neatly fit into this picture. So you've received a genetic testing result, presumably, as a member of the audience. And when we think about when you receive these genetic testing results, when this laboratory is first flagged and you've sent the sample off to the laboratory, they're reading through the genes, they're looking for these genetic changes, the laboratory has to come to a consensus about whether a particular genetic change may be disease-causing or is not disease-causing. So there's a very standardized way that this is done using particular sets of evidence, such as if a variant is present in the general population, if the variant has been seen before in other people with this condition, and things like that. And then they use this information in order to classify a variant into one of these 5 categories: pathogenic, likely pathogenic, uncertain significance, likely benign, or benign. If a variant is thought to be likely benign or benign, it won't make it onto the genetic report because it's not thought to have implications for that individual's health. So these are the different classifications that will end up on our report. So a pathogenic variant means that this variant is definitively disease-causing. There are multiple lines of evidence that support that this variant is indeed causing the disorder. Whereas a likely pathogenic variant means it's very likely the cause of disease, but there's just not as much evidence as there is for a pathogenic variant. And so that's why this distinction is made, although typically we think of these variants as diagnostic. And then the last way that variants may be reported on your genetic testing results are what's called a variant of uncertain significance, which means that it's unclear what role the variant is playing in the disorder. There's not enough evidence to say one way or another, is this variant disease-causing or not? And so it's important, if you get back this type of result, to discuss this with your healthcare provider in order to better evaluate the role of this variant in your child's condition, as well as what next steps may be taken to resolve this uncertainty. So your genetic testing results should always be interpreted in a clinical context. I think a good example of this was the question that we had earlier regarding whether a variant was gain of function or loss of function. And then based on clinical presentation, we can make a good guess there, or an informed way of thinking about it. So we always have to think about the genetic testing lab's assessment of a variant and then your clinical team's assessment of a variant, and then those come together to best understand the role of this variant in your child's condition. And then this information can be used to say, this is definitely the diagnosis, or maybe there are some features that require additional testing. So what does this all mean practically? If a variant is classified as pathogenic or likely pathogenic, we would consider this a diagnosis, and then no further genetic testing is needed as long as we feel that all features that are present in that individual are well explained by that particular gene. And then if a variant of uncertain significance is present, it's important to discuss this with your care team and discuss what additional testing may be needed to better resolve whether this variant is causative or not. Some examples of things that are commonly done are parental testing. As we discussed, most KCNQ2 variants are what we call de novo in the child, meaning their parents test negative for that genetic change. And if that is the case for a variant of uncertain significance, that may make us more suspicious. And this actually may change the classification of the variant on the laboratory's end as well. And then also more research-based testing. If we do functional studies of a particular variant, that may make us more suspicious or less suspicious of a given variant, but this is usually not used within the clinical laboratories. So some general thoughts about genetics. There are some genotype-phenotype correlations, meaning relationship between a given genetic change and the symptoms that we may see in multiple children with that change. But the other variants have more features that are variable. And kind of the way that we think about a genetic diagnosis, it explains the symptoms that we see in that child, but it does not necessarily predict. It gives us some idea of what to expect in the future, but really, as I'm sure you all know in this audience, every child is different, everyone is unique, and we use symptom-based care and assessments as well as the genetics to create an overall picture to think about the future. So in summary, the most common types of genetic tests that people have in their workups that may identify disease-causing changes in KCNQ2 are chromosomal microarray, gene panels, or whole exome sequencing. There are many different types of genetic variants in KCNQ2. So especially in this era of precision medicine, it's really important. Know your CDOT, know your PDOT, know what particular variant your child has, because this will become increasingly important when we think about gene or variant-specific therapies. And if you have a variant of uncertain significance, this should always be interpreted in a clinical context to determine if that variant is suspicious, not suspicious, or whether further testing may be needed. Thank you all for your time. I work in a much larger group at CHOP with a large group of genetic counselors, neurologists, and more that I can even name. So if you're interested in coming to see us, please feel free to reach out. And otherwise, thank you so much for your time.
Thank you so much, Stacy. We have time for a couple questions. Does anybody have a question that they'd like to ask? Or Claire, is there anybody from online? You? Okay. Can you come on over to the mic, or if Jim has it otherwise?
Thank you. Just a quick question, because we talked a lot about the epilepsy genes, but Elliot, my son, for example, also has autism. And I know there are genes that are more associated with autism. Have you looked into the correlation between those two? Because obviously he wasn't then, we didn't rerun a panel for autism specifically. He was assessed and diagnosed clinically. But is there any studies or anything in terms of looking at that correlation or what the outcome might be, depending on the genes that are associated with epilepsy and autism?
Yeah, that's a really good question. So when we think about all of these different neurodevelopmental conditions like autism and epilepsy, they're really more like a Venn diagram. There are many children with autism who go on to develop epilepsy, or with epilepsy who go on to develop autism, depending on the timing of things. So when we think about these gene panels, epilepsy gene panel, autism gene panel, these are kind of arbitrary distinctions. And if you order them at one laboratory versus another, it will contain different genes. So it's not like a medical test where they're looking at, for example, blood glucose or something like that. They're really defining what they're looking at. And so a lot of these genes can cause both epilepsy and autism. So they are kind of arbitrary distinctions, and often these panels have many overlapping genes.
Do you have a follow-up, or are you good?
But then does that mean that it might be multiple genes that are implicated in kind of that diagnosis? Because we only have KCNQ2, and I understand that KCNQ2 can, have epilepsy and autism, but is there a chance or a risk that there's another gene that is causing the autism? And would that impact, the outcome?
Yeah, that's a good question. So We do have children and families who have multiple genetic diagnoses, and I would say the threshold to consider additional workup or looking for alternate etiologies would be if the symptoms that are seen in your child are felt to be out of proportion with what we know about that gene. Or if they have additional symptoms that don't fit well with a particular genetic diagnosis, that's when we would think about sending for additional testing and looking for alternate etiologies.
Anybody else? All right. Well, this is, thank you so much, Stacy. I really appreciate it. If you have further questions, you can also chat to Stacy individually. That's the beauty of being here in the room. We're also equally thrilled by all of us who've joined online, and a special shout out to Kara Bolter, who actually reached out to Stacy and brought us the team from CHOP. Unfortunately, her beautiful boy, Nolan, isn't well enough, so she wasn't able to be with us here today, but she also helped to arrange the fact that we have those individual one-on-ones for some of the families who signed up for that ahead of time. So very big thank you to Kara and to everyone who's helped us out. And when I'm doing thank yous, I wanted to say a few more. Big thank you to Xenon. They've given us a huge amount of money to help hold this conference. We would also like to thank Biohaven and Invitae. Putting on conferences like this is expensive, and we really do appreciate you giving us that financial support and making it possible for all of us to be here today. And I don't know if anybody's had a chance, because we are here in the room, to have a look at this online, but it looks beautiful. So Jeff Cooper and the team at Syndicast, thank you very much for doing such a great job. We appreciate you all as well. We're going to take a break now for lunch. Can everybody please come back around 10:30? I think it's just right here. I'm saying lunch, I mean morning tea. Can you take, it's a little early. You know, I don't even know what time it is in Australia, and I refuse to look at my clock for that. So apologies if I've got the times wrong, but we'll have a little break for morning tea. And if you can be back by 10:30 or a little bit after, we're staying nicely to time, and we'll try to keep up on that. And we'll see you back in a few minutes. Thank you very much.